Hey guys, it's Dr. Mason here, and Dr. Mick again is holding the cell phone for this video. Today we will be going over a lamprey. So lampreys are jawless fish. They are a very primitive fish in the taxon Agnatha. So what does that actually mean? It means, if we look at it taxonomically, we're going to be in kingdom Animalia, that's where we are, phylum Chordata, that's where humans are too, subphylum Vertebrata, that's where humans are too, but infraphylum, they are an agnathan. So again, a jawless fish, very primitive, because almost all vertebrates actually have some type of jaw. The evolution of vertebrates in general usually has to do with their skull. Skull evolution actually has to do with their jaws, which we will talk about in lecture. But it is a protovertebrate, meaning that all vertebrates that we know of today that are currently extant, that's the opposite of extinct, originated from protovertebrates, early primitive vertebrates, early primitive fish in this case, like the lamprey. There's currently 40 species of lamprey that are extant as well. And if we want to break those 40 species of lamprey down into different feeding functional groups, there's going to be 3 different types of feeding functional groups of lamprey. The first one are filter feeders. So lampreys will actually take their buccal cavity right here and latch onto a rock just like that. And then they're just going to filter feed with the microorganisms that's in the water column. We also have flesh eaters, quote unquote, of lampreys. They do the same thing. Instead of my hand being a rock in this case, they're going to latch onto the back of a fish, usually where it's a little bit more meaty and some more muscles towards the back of the fish. And then they're going to use this tongue, which we'll actually get to. Maybe Maria could zoom in on this tongue right here that actually has 3 teeth on it. They'll latch onto the fish, have that tongue protrude out and start digging into its host fish and start eating that meat. And then the third type of feeding functional group of lampreys, they're bloodsuckers. So it's more or less the same thing. The lamprey is going to latch onto a fish, usually the belly of the fish in this case, and then use that tongue again to start protruding out and feeding on blood. So a little bit about lampreys. Very amazing animal just because of how old their lineage is. So let's go over some external structures, and then we'll eventually go over some internal structures. But external structures, the easy parts. Eyes. Here's one eye right here. It looks a little foggy. If it was still alive and not preserved, it would actually be clear. And just like us, they have 2 eyes. So here's the other one. They also have gill slits. So these are the external gill slits. We'll be looking at the internal gill slits once we dissect the animal. They should have 7. Let's confirm this. 1, 2, 3, 4, 5, 6, 7 gill slits right there. When folks go to the aquarium and actually see one of these things alive, they actually think it has 8 eyes because they look at each one of these gill slits and thinks it's an eye. But it's not. Hopefully you guys know that by now. What else do we have? If we look at dorsal of the head of the animal, we see this little opening right here. This is the animal's, the lamprey's nostril. Well, it is a nostril, but the specific term that you guys need to know is it's a long one, nasohypophyseal opening. Nasohypophyseal opening. Say it out loud right now to yourself. Nasohypophyseal opening. But again, it's basically its nostril. Every lamprey, you're going to also see this white area right behind its nasohypophyseal opening. And this more or less is its pineal glands. And I want you guys to look up what is the function of a pineal gland in general, but also specifically to the lamprey, okay? The other external structures that we actually see, it's the entire trunk of the animal. So the trunk of the animal is basically its body. We're not counting its head. We're not counting its tail, which is towards the end here. This entire section right here is its trunk, okay? Other external structures that I want to go over are the dorsal fins. So here are the 2, lampreys have 2 dorsal fins. Dorsal means the top, so here's one right here. This is going to be anterior, closer to the head, the anterior dorsal fin, and here we actually have a posterior dorsal fin. These are considered single fins, not paired. So what do I mean by that? I mean, if you think of a fish, right now, it will have bony fish. Most fish species, as we know, have paired fins. So this anterior dorsal fin would also have a paired fin underneath here in more recently evolved fish versus primitive fish, okay? These lampreys, they swim like a snake underwater because they only have dorsal fins that are single versus paired. Paired fins evolved eventually to maneuver better in water, okay? The other external feature that we're actually going to see is towards the ends. This is the caudal tail, or the caudal fin, which is by the tail of the lamprey. And this is how it pushes forward. It's basically its propeller as it's swimming underwater, okay? What's really cool, I find very fascinating, there's something called the lateral line system. Many animals have this, including non-aquatic animals. Most aquatic vertebrates have this though. I hope the video could actually show it, but you'll see this animal has all these little bumps on its side. Here's some bumps here too, but I'm looking actually through the video now that Dr. Mick is actually showing. So these are the lateral line system. I call them like little white bumps. I want you guys to think of, what do you think the function of these bumps are for a fish? And then look it up to figure out if you're right or wrong. So make a hypothesis. Why do you think this lateral line system is intact with this lamprey? Okay? If we stay ventral under the animal, let's go towards this opening towards the tail. You guys should be familiar with this, that it is a cloaca. Cloaca, all-purpose chute. So you should know what material are going to come out of this cloaca for this animal. But there's also this structure right here, this microstructure I'm lifting up. This is the urogenital papillae. So make sure you also look up the function of the urogenital papillae. That's a lot of the external structure that we're actually going to talk about in this video. But lastly, technically, it's going to be an internal structure. We're actually looking at its buccal funnel or buccal cavity. So there's three structures that I want you guys to know. Again, technically, these are internal structures. But around this buccal funnel, keep in mind, that whole thing is the buccal funnel where the fish latches onto a rock or host fish. But all along here are minute papillae that are around the animal's mouth. And they're designed for a few things. One of them is to actually detect different micro vibrations in the water, but also to help the animal latch on to the host fish or the rock that it's going to filter feed on. Then we actually have its own teeth, which are all around here. Super impressive teeth, to be honest. And then we also have the tongue, which is right here. Is it shown in the video? Yep. So here's the tongue, and keep in mind the tongue has 3 individual teeth, and that tongue, again, will kind of shoot out like this to start feeding on whatever fish host that it's attaching to. And the 3 teeth there are a lot thicker than these teeth right here to help the animal start digging into the flesh of its host fish. Really cool looking animal, potentially scary looking animal. But again, we humans and all vertebrates that we're thinking of that are in existence today evolved from proto-vertebrates just like this lamprey.
Hi guys, this is Dr. Mick here. Dr. Mason is behind the camera this time. So we're gonna do what's called a sagittal cut on this lamprey. So when we're doing a sagittal cut, we're gonna cut along the dorsal, which is the back of the animal, along the midline. So that way when we have our cut, we'll essentially be putting it into 2 halves, like if you were opening a sliced hoagie, for example. So we're going to be as careful as we can, but sometimes when we make these cuts, we don't get perfectly on midline. It's also an extra challenge with the preservatives that are here because sometimes things are preserved in a slightly abnormal position. So what we're gonna do is it'll probably be good if you have a couple of your group members holding the animal, so that way we're holding it in place to limit our, thank you, Dr. Mason, limit our chance of going off midline. And then I would advise holding with your non-dominant hand at to the part of the animal where you're about to cut. You want to take your scalpel. When you're using your scalpel, you want to be very careful, but good handling. You want kind of a finger on each side of the handle, and you have more control over your blade, and you're less likely to hurt yourself if at the top of your handle, the dorsal part of your handle, you keep your finger there. So you want to, again, be very careful, but we're going to go along dorsal midline. When you're using these structures that Dr. Mason just talked to you about, that can really help guide you, as well as going along where the fins are will help you as well to stay on track. It's not going to be perfect, and don't stress too much, just try your best. It is a little extra challenging to cut around where the teeth are, just because those are a pretty firm structure. But if you press downward with your scalpel, that can help. Generally, we want to do a smooth slicing motion, but you do need to dig down a little deeper there when you're doing the teeth. But you can see how it's opening up, again, like we're opening a sliced hoagie. So we're just gonna go down. Better to be slow and be as accurate as you can. And even if you just slice along the top of the animal and don't get all the way through, you can always go through later once you see where you're at. So we're just going slow here, trying to get everything as straight as we can. You can always reposition as you go on, so you move your fingers to hold the animal steady, which should make your cut a little better, and will also be a little bit better for decreasing your risk of injury. We're working our way towards the back. I'm going to reposition our friend again. Hopefully this will be a good cut. I'm hearing a crunching. All right, and we should be about past the cloaca here. So at this point, we don't need to worry too much about the tail region. So let's see what we got. So we're gonna open this guy up, and I would say it looks like we have a pretty good cut here, although not all the way through in this region. So we're just gonna make that a little bit more exact. So I'm gonna first push down on either side so we can maximize the cut that we did get. And then we will just kind of cut along our line here too. Cut that as best we can. And you really only have to go down about this far once we reach the end of the structure, which Dr. Mason will go over with you in the next video. We don't need to worry about too much past that point into where the tail would be. All right, so in this next video, then we'll talk about what we found here.
Hey guys, so here we are with a beautiful sagittal cut done by Dr. Mick. I have my lab assignment to my right off screen that should be more or less the same that you guys actually have. So let's go over some of the structures that I'm seeing right now for the sagittal cut. You guys should know, here's the buccal cavity. So here's the opening right here of the mouth, essentially. Here we actually have some buccal muscle helping this lamprey like latch on to whatever host fish or rock that it can latch on. So if we go down this buccal funnel or the buccal cavity a little bit, we're going to run into the pharynx, kind of right into this area right here. Eventually, the pharynx is going to turn into the esophagus, all somewhere around here. And it's a small lamprey that we're actually dealing with, this species, so it's kind of hard to see from the pharynx to the esophagus. But if we follow the esophagus all the way down, we will eventually get into the intestine. So here, I'll see the intestine mostly here. This what I'm lifting up right now is the intestines that go all the way from the buccal cavity all the way out, actually to the cloaca, which would be, it's harder to see now because we actually, I'm trying to look for it, because we actually did a sagittal cut. But yeah, as you guys can imagine, cloaca is the all-purpose chute, so all the digested food that's going through the intestine at this point is going to come out of the cloaca. Keep in mind, this animal, the lamprey, only has one intestine. There's no small intestine, there's no large intestine, and one thing that's very obviously missing from the digestive system of this lamprey is, think about it, the stomach. There is no stomach. So the lamprey is eating something, it's basically going into the pharynx, going into the esophagus, and going right into the intestine, okay? Quickly, because we're down in this area, so the intestines, we could actually see some of the gonads. So here are some of the gonads of this animal. Hopefully you guys know what gonads are, okay? Also that we're in this area, so normally, in my experience, in Dr. Mick's experience, the intestine isn't colored green, but in this case it's colored green because the liver, in our experience, normally is green. So here's actually the liver, well, with the intestines. Hopefully you guys could see that on the video. Intestine and liver. This whole structure right here is the liver, okay? So going back up anteriorly to this animal, right here we actually have the brain. It is not this, we will talk about this later, but the brain is going to be right here. Unsurprisingly, the brain is going to be attached to the spinal cord, which is all along this area right here and all the way down the animal. Ventral to this spinal cord is actually the notochord, which is right here. And if we haven't talked about it already in lecture, but remember, this lamprey is a protovertebrate. They do not have a true vertebrate or a true spinal cord, even though that I'm talking about a spinal cord right now, as we know it, that has individual and different types of vertebra. We evolved our spinal cord, our true spinal cord, and our different vertebra from primitive protovertebrates like this. But brain, spinal cord, a very primitive spinal cord, and eventually the notochord. Said eventually because at some point in evolutionary history, these two combined together. Okay? We also have myomeres. So we have dorsal myomeres here. Myomeres is a fancy way of saying muscle. And here we have our ventral myomeres, our ventral muscles of the animal, helping this fish mostly swim through water, looking for prey, looking to mate, different things that animals do. We also have our heart, which is right here. Hopefully you guys know the function of the heart. And you guys also have on your work assignment the pericardial cavity. So the pericardial cavity is where the heart's actually located. Here's the other side. There's your pericardial cavity. And just for funsies, I'm looking upside down. Here's actually the liver right here. And you guys tell me, or you guys know, you can't tell me right now, what this structure is and what this structure is. Hopefully you guys know, okay? So we did heart, we did pericardial cavity. Let's go now to the pineal gland. So if you guys recall, the external structures is this white kind of structure, the dorsal part of the head. So now that we did a sagittal cut, we actually could see the dissected version, the sagittal cut of the actual pineal gland, okay? Pineal gland right there, our brain is right here, but now let's actually talk about this. This is the olfactory bulb. You guys look up what is the function of the olfactory bulb. Keep in mind, the olfactory bulb is attached to the nostril or the nasohypophyseal opening. Say it again out loud to yourself. It's a long word, probably a new word for you guys. okay? But we have all these things right here: the olfactory bulb, the pineal gland, and also the brain, which is attached to the spinal cord, okay? Last but not least, what I have for you guys, internal structures, is our internal gill slits. Do you guys remember how many gill slits that this animal has? 7. Hopefully you guys answered that, or at least thought about it. But the gill slits might be easier to see, the internal gill slits on this side right here of the sagittal cut. But we can actually see that here's one opening of a gill slit. Here's another gill slit right here. And then here's another gill slit right here. And I could also, I'm not going to do it, but stick my probe through these external gill slits, and it undoubtedly will come out through the internal gill slits. Okay, so when you guys do a sagittal cut, keep in mind you have 2 pieces to work with. If you guys have a larger group, maybe of 3 students, you guys could play around individually or as a group with the 2 different sides. One side is probably going to be better than the other side. Again, we could see the pericardial sac cavity right here. The heart's on this side. This side has the intestine. Remember, it's one intestine. This side does not have the intestine. This side had a better examples of internal gill slits, but play around with it, have a good time with these specimens, and remember, you're watching this video right now, and you might be thinking your specimen is going to look exactly like our specimen. But if you guys remember the frogs, that's not always the case. So orient yourselves, find a structure. For example, if you could find the olfactory bulb, which is relatively easy, you have an idea that the pineal gland is going to be here and the brain is going to be there as well. So orient yourselves, watch the video, read your lab manual, do what you need to do to make sure you know these structures and you could say these structures and properly spell these structures, because I'm trying to prepare you as best as I can for the practical. Okay?
Hi guys, this is Dr. Mick again. So now we're going to be doing some cross-section cuts. Now, you will need a second specimen for this because your first specimen was where we made those sagittal cuts. So where we did the sliced hoagie cut. These cross-section cuts are going to be more similar to if we were chopping a cucumber, except we're going to be more precise and do fewer sections. We're only going to cut 4. So again, you're going to get a new specimen. Some of these specimens do have some mold growing on them. On this guy, we can see that there's a little along here. So I would just say for these, use your moldier specimen for the cross sections, just since you're generally gonna have to handle it a little bit less. So on page 22 of your lab manual, we see an area that talks about the cross sections that you're gonna do. So we're gonna make 4, as I said earlier. So one cross section is going to be through the pineal organ and the eyes. So we'll do that in a minute. Your second cross section is gonna be between the 3rd or 4th gill slit, about mid-length along this branchial region. So we'll count our gill slits to make sure that we're going in between. So 1, 2, 3, 4, 5, 6, 7. So we'll go in between the 3rd and the 4th gill slit. And then the 3rd cross section is the part of the trunk with the liver and intestine. So it is a little difficult with how these guys kind of shape into curvature due to the preservative. So we want to straighten them out. And again, someone else in your lab group could be really beneficial to hold this specimen to keep it straight. But sometimes what I like to do to make sure that I'm not cutting too anterior or too posterior would be to just compare it to my other one. Now, of course, they're not all the same size, but when I look at this, for example, and we see our 7th, which is our last gill slit, since we know that they have 7 gill slits. And if we flip that over, we can see that that lines up with the pericardial cavity. So I know that when I'm making the cross section where I want the liver and intestines, I'm gonna go a little bit lower. And that will still give me the liver and the intestine, but I'm not gonna be cutting into that pericardial cavity. And then last but not least, we cut through the tail, a cross section through the tail. Now, when the company injected these guys with dye, they already made a cross section near the tail. But when I look closely, I can see that after the spot where they made that cross section is where we have our cloaca. So I am going to cut further back past the cloaca to make my final cross section of the tail region. I hope that all makes sense. So now let's go about making our cross section cuts, and we'll see how it goes. So move these other guys out of the way. And our first one, as we can see on page 22 of our lab manual, our first cross section is through the pineal organ and eyes. So I want to line up, and I see we've got our pineal organ, we have our eye. So we're going to make a cross section cut right through there. So again, we're going to think of like chopping a cucumber for this, and we're just going to try and make a nice straight cut. Most of these scalpels are pretty large, so if we just line it up central, we can just hopefully make a pretty smooth cut there. And let's see what we have. There we go. We'll be identifying these structures in the next video. But we have those. If you have any remaining tissue along the bottom, that's no problem. You can just kind of cut that, thin layer of attachment there. So just be gentle. And now we'll work on our second cross-section site. So that's where we want to go between our third and fourth gill slits. So again, let's count, make sure. 1, 2, 3, 4. So we think we're in there. 5, 6, 7. So we're gonna cut right in this spot. And I'm just gonna keep my finger there while I get my scalpel together. Obviously move your finger before you're going to be using your scalpel blade. And we're just gonna try and make a nice, as straight as we can without doing too much of a sawing motion, just so we don't harm any of the structures we're gonna be looking for. If there is one that you do happen to really kind of butcher, that's okay. You can just then make another smaller one a bit further down. Here we go. That's beautiful with the dye. That's a really lovely view of those gill slits. So next we have the part of the trunk with the liver and the intestines. So if you can remember from what we just talked about, if I were to cut right by the 7th gill slit, that puts us right in touch with the pericardial cavity. So I'm just gonna go a little bit further down. We don't wanna go too far down or we'll miss most of the liver, but I'm gonna cut about here and we'll see what it looks like. If we don't like it, then we'll just make another one. All right. Let's see what we have here. All right, I can definitely see that we have our liver because we see that green area. So I think that will be good. We'll confirm in our next video when we identify these structures. And then last but not least, we need the tail, which is generally one of the easier cross-section cuts. Now, since the tail is so thin and we have this dorsal fin, cutting up top of this way can be a little more difficult. So it's okay for this one if you lay it on the side and just try to make a pretty good cut there. And so we got the most important parts. I just need to cut the rest of our fin, so that part's easier. There we go. Let's see what we have for this last one. All right, these are looking good, but we'll find out for sure very soon.
Hey guys, so we're going to go over the different cross sections that Dr. Mick beautifully did. For whatever it's worth, I am going to go backwards in the lab manual. What do I mean by that is, it's that there's 4 different cross sections, but I'm going to start with 3.6D first, which is the tail cross section. Why I'm doing that, it's because the tail, it's the easiest for all the structures. So I want to get you guys off to a good start before we get to more structures. So the first structure that I want to talk about that's on our lab assignment is going to be the fin ray, which is right here, basically the fin. We also have the spinal cord. The spinal cord is going to be this opening right here in our cross section. We also have the notochord, which is right here. We have the caudal artery, which is going to be this very, very, very tiny opening right there. Looks like Dr. Mick is finding the opening, which is going to be here. The larger opening, ventral to that, is going to be our caudal vein. So caudal vein is the big opening right here. Caudal artery is going to be dorsal of that, extremely tiny, right above the caudal vein. Keep in mind that veins in general carry deoxygenated blood going back to the heart, and arteries generally carry oxygenated blood. We'll go over that more when we go over circulatory system in depth. So again, this is the tail cross-section, 3.6D. So now let's go to the other cross-section. This is liver and intestines, 3.6C. Getting a little focus there. Okay, so here we again have our spinal cord, which is right here, that opening. We have our notochord right here. We have our posterior cardinal veins, which are the beautifully dyed, these blues right here. Posterior cardinal veins. Remember what I said about veins, about them actually carrying deoxygenated blood back to the heart. This red right here is the dorsal aorta, also beautifully dyed. The aorta is a type of artery. We also have all this green area here, which is our liver. And this black structure right here is our intestine. So remember, one intestine. We're getting on the focus again. I will stop moving to let Dr. Mick focus. Right here is our intestine, right here is our liver. But also in 3.6C, figure 3.6C, you should be able maybe to find the gonads. In our cross-section, our gonads are hidden by the liver, but see if you guys could find your gonads as well, okay? Hey guys, so let's go over the structures now on figure 3.6B, the cross-section of the branchial slit. Here are myomeres. So these again are the muscles of the animal, of the lamprey in this case. We also have our spinal cord, which is right here. We have our notochord, which is right here. We have our anterior cardinal veins here and here. Dorsal aorta right there. We have the esophagus, which will be this opening right under the dorsal aorta. I'm trying not to damage the specimen too much, but it's a little opening right here, again under the dorsal aorta. We have the pharyngobranchial tube, which is right here, another opening, which is going to be in here. Then we have the ventral aorta, this blue circular structure right here. The inferior jugular vein, it's actually going to be right here. There's going to be an opening right here, again, inferior jugular vein. The lingual muscle is right here. We have gill filaments on the left side and also the right side, which I find really cool is the gill pouches, so you can kind of open that up a little bit. And that's it for figure 3.6B, brachial slit. Hey guys, here is our last cross section. So this is going to be 3.6A, the pineal gland. So our first structure is actually the pineal gland, which is right up here. The lab manual says pineal organ. I use pineal gland and pineal organ synonymously for whatever it's worth. The next structure we actually have is our nasal sac, which is going to be right here. Also, our nasal sac, keep in mind, is also called the olfactory sac. The next structure is the hypophyseal pouch, which is this opening right here. Then we have carotid arteries, which are nicely and beautifully dyed. I think Dr. Mick and I are spoiled with this specimen so far with the veins and the arteries. Our next structure is the lens of the eye. So the lens of the eyes here, here, this black area right here is the actual eye. So here's the black is the actual eye, but the lens of the eye is the white material that's in the center of this black, which again is the eye. We have the buccal cavity, which is this large opening right here. Hopefully you know where the buccal cavity actually goes out to. And then we also have the piston cartilage, which is right here, the piston cartilage. And last but not least, again, we have our myomeres, which are the different muscles. Dorsal myomeres and ventral myomeres. All right?